Let's start with the basic idea of an antigen. Think of it as a unique marker on a pathogen that our immune system can recognize. Antibodies, which are like targeted defenders, specifically bind to these antigens. This binding is crucial because it tells our immune system exactly what to attack. So, the key thing is that antibodies don't just bind anywhere, they recognize very specific parts on the pathogen, helping our body identify and fight infections effectively. Now, what exactly is an antigen? Simply put, it's any molecule that can trigger an immune response. That means it can activate our defenses. But it doesn't stop there. Antigens also specifically bind with molecules like antibodies or immune cells such as lymphocytes. Interestingly, the word antigen comes from antibody generator, highlighting its role in prompting antibody production. This sets the foundation for understanding how our immune system detects and reacts to threats. Here's an important distinction. Not all antigens trigger an immune response. Some antigens are recognized by immune receptors but don't activate the system. These are simply called antigens. On the other hand, immunogens are a special class that actively induce an immune response by turning on B or T cells. So, while every immunogen is an antigen, not every antigen is an immunogen. This difference helps us understand why some molecules provoke immunity and others just get noticed without action. Let's delve into antigenic determinants, or epitopes. Our immune system doesn't recognize antigens as whole molecules because they're usually large and complex. Instead, it focuses on specific regions called epitopes. These are the immunologically active parts that lymphocytes detect. Chemically, epitopes can be made of sugars, amino acids, or hydrocarbons. Also, antigens can have one or multiple epitopes, monovalent or multivalent, which influences how many immune responses they can trigger. Understanding epitopes is key to grasping immune specificity. Several factors influence how well an antigen triggers an immune response. Larger molecules tend to be more antigenic because they're easier for the immune system to detect. Foreignness matters too. If something looks very different from our own molecules, it's more likely to be recognized. Chemical complexity and stability also play roles. Complex, stable structures tend to provoke stronger responses. Interestingly, the biological system varies. What's immunogenic in one person might not be in another. Finally, the amount and route of exposure affect antigenicity, with very low or very high doses being less effective. Now, let's explore the key types of antigens. Complete antigens can induce antibody formation and cause visible immune reactions. A hapten is a substance that can combine with a specific antibody but lacks antigenicity of its own. T-cell dependent and T-cell independent antigens differ in whether they require T helper cells to produce antibodies. Superantigens activate many T cells non-specifically, which can lead to intense immune reactions. Self-antigens are molecules on the surface of a cell that the immune system recognizes as belonging to the body, rather than foreign invaders. Heterophile antigens can react across species due to common structures. Sequestered antigens or hidden antigens are typically not encountered by the immune system during tolerance development. Examples include eye-lens protein and sperm. Neoantigens are altered molecules that trigger responses, often seen in mutated cells. Human erythrocyte antigens form the basis for human blood groups. Histocompatibility antigens, or human leukocyte antigens, HLA, are cellular determinants specific for each individual within a species associated with the plasma membrane of tissue cells. Lastly, tumor antigens are unique markers on cancer cells that the immune system can target. Adjuvants play a fascinating role in immunology. They don't change the antigen itself, but enhance its ability to provoke an immune response. They do this by improving how macrophages process the antigen, extending the time the immune system is exposed to it, and boosting the growth of necessary immune cells. Examples include ammonium salts, emulsions, and liposomes. So, adjuvants basically act as helpers, making vaccines and other immune triggers more effective without altering the original molecule. Finally, let's talk about the paratope. This is the specific part of an antibody that binds to an epitope on the antigen. Think of it like a lock and key. Each paratope fits uniquely with its corresponding epitope. This precise interaction ensures that antibodies target the right part of the antigen. The diagram illustrates how different antibodies can bind to different epitopes on the same antigen, highlighting the specificity and versatility of our immune defense system.